Our first panelist is Subhra Priyadarshini, who is the chief editor of Nature India. She has worked as a journalist with several Indian dailies. She has received the BBC World Service Trust Award and the Panos South Asia Media Excellence Award. Through Nature India's many workshops and outreach activities, she trains scientists, healthcare professionals, and journalists in science and health communication. Welcome, Subhra. Our next panelist is Dr. Anurag Agrawal. He's been introduced once earlier, but for those of us that are new here, he's the Dean of Biosciences and Health Research at Ashoka University. He's a trained physician, but is also a scientist and focuses on the intersections of biology, medicine, and data. He's been lauded with several awards. The WHO Technical Advisory Group for Viral Evolution he's chaired. He's received the Shanti Swarup Bhatnagar Award and the Sun Pharma Foundation Award in 2020. Welcome, Dr. Agrawal. Our third panelist is Subash Lingareddy, who is the co-founder of Mapmygenome. So a few of you may have attended the booth that Mapmygenome has over there at the STEM booth counter. Now, Mapmygenome is a molecular diagnostics company that leverages state-of-the-art DNA sequencing and genotyping technologies to screen for genetic predisposition and diagnose complex genetic disorders. This company is revolutionary in healthcare in that it enables people to take proactive steps to safeguard their own health. Welcome. And our final panelist of the day is Dr. Debojyoti Chakraborty, who is a scientist at CSIR-IGIB. And a little bit before that, he enthralled us with a mesmerizing sitar performance, which I hope a few of you had the opportunity to attend. He studies novel factors that modulate cellular pathways using cerebral organoids. He has been awarded the CSIR Young Scientist Award in 2020 and also became one of the 30 EMBO Young Investigators in the same year. And besides his academic interests, he is an acclaimed sitarist, like I just mentioned, and he has performed widely in India and abroad. He has won a national scholarship in instrumental music from the Ministry of Culture, the Government of India, followed by several key mentions and awards, such as the State Music Academy Award for Best Instrumentalist in 2007. So we've got a very diverse, esteemed panel with us. Let's welcome them all once again. And we're really looking forward to this, and I'll hand this over to Subhra.
It's a change of plan, like we said earlier, in the spirit of having a conversation with you all more than conversing with the panel itself. So we want you all to get closer, and we came down from the podium just so that we can have more interaction. So many, many thanks for arranging this lovely panel discussion, which has luminaries, as you can see, from all kinds of spaces in gene editing, stem cells, and personalized medicine. So in the spirit of the festival, of course, I'd like to begin this panel without giving too much of an introduction, but starting it with a poem which is around gene mapping. So many of you will relate to what a geneticist undergoes in his lifetime while mapping a genome, which is the next big thing in biological sciences, of course. So let me just begin with British poet Michael Simmons Roberts' poem, who uses a very specific and lengthy metaphor, if you will, to liken the mapping of the genome to a very bendy, winding, and occasionally uneventful road trip, which geneticists here will affirm to. The road that he suggests here in this poem is equivalent to the winding helixes in our DNA. So here goes. Mapping the Genome by Michael Simmons Roberts. Geneticist as driver down the gene codes in, let's say, a topless coupe, and you keep expecting the bends. Real tire testers on tight mountain passes, But instead, it's dead straight, highway as runway, helix unraveled as vista, as vanishing point. Keep your foot down. This is a finite desert. You move too fast to read it. The order of the rocks, the cacti, roadside weeds, it's all a blur to you. Every hour or so, you pass a shack, which passes for a motel here, tidy faded rooms with TVs on for company, the owner pacing out his empty parking lot. And after each motel, you hit a sandstorm, thick as fog, but agony. Somewhere out there are remnants of our evolution, genes for how to fly south, sense a storm, hunt at night, how to harden your flesh into hide or scales. These are the miles of dead code. Every desert has them. You are on a mission to discover why the human heart still slows when divers break the surface, why mermaids still swim in our dreams. So in essence, this poem, basically, though we may have succeeded in mapping genomes of all kinds, and subsequently are able to make nicks and cuts for our own good or for whatever reasons we want to make those cuts, this poem basically says that some long-held secrets may always remain a mystery to us. So this sets the tone beautifully for our panel discussion today, because we're going to not just talk about the science of genetics, but also how it interlaces with social norms, society, the enigma of all of it, the ethics and the impacts that future generations may be facing from gene editing. So most of the ethical discussions that we have today around gene editing, and since this is a mixed crowd, we'd perhaps focus on the human genome more, and because of the fact that the ethical discussions actually center more around the human germline, and because editing changes made in the germline will have impacts on the future of humankind. So let me begin by asking Anurag here about a more wider sense of what gene editing, a broader overview of what gene editing is, what the trends are in the science itself, and in stem cells, of course, and why increasing scientific and technological feasibility must be accompanied by a more social science perspective.
Thank you so much. And I think our coming down to earth from the higher podium of science is a good metaphor to keep in mind as we discuss the importance of intersections. So in your poem, you saw the tire pressing bendy roads that didn't come by. Instead, patches of straight, slightly vined, with some obstructions from time to time. On the science side, there are always three parts. A, given a sequence of a genome, do you know what it does? In case you were able to figure out a change you could make to make it do what you want it to do compared to what it is currently doing, is it technically feasible? When you say technically feasible, you have only two questions. Can you introduce the change that you want? And can you not introduce other changes that you did not want? Then the last part is to verify the change that you wanted to make actually did what you wanted it to do. Now, when it comes to disease with single mutation, sickle cell, we just heard Dr. Bang speak about sickle cell disease. You can identify specific mutations that if you change, you will change the property of the cell. If it happens to be a liquid tissue that you can destroy and put back in again, you have a way of changing the entire human's life from that point there on. Obviously, it gets complicated in other tissues, which you can't just take out and put back. The bendy part comes on that last part. What was it that you were intending to be doing to improve health? And at what point do you start distinguishing between disease and other things? Because of the wonderful world of genomics, and I spoke about the fourth wave being an intersection of the genomic revolution, we also have very interesting mutations. We have mutations that prevent HIV infections of T cells. We have mutations that can give stronger humans. Of course, we know some mutations for color. But we know other mutations that will lower your cholesterol. So Helen Hobbs found PCSK9, a mutation that naturally is found, lowers your cardiovascular risk, reduces your cholesterol. In monkeys, done once in the liver, gives sustained lowering of cholesterol for 3 years by now and reduce atherosclerosis. Then the question comes, for what all things do you want to deploy this therapy? Do you want to consider ways in which you introduce this into the germline of the human so they're, after proving it is beneficial, after all the proof is done, do you want to go germline? Do you want to create a branch of the human race which is, in one sense of the word, better? That's where the tires are going to press on the bends. Because natural science, there is a backing to it. Social science is about us. And knowing ourselves is the hardest thing because we are not static. Something that would have been considered acceptable 100 years ago could be called eugenics today. So I think those are the big challenges, and I'm not really a geneticist, so much more qualified people will come after me to discuss this point.
Sure. Thanks for that analogy. And of course, it reminds me of the famous saying by American psychologist Albert Maslow, who says, if you have a hammer in your hand, everything looks like a nail, and you want to hammer on at whatever you want to. So that's a great way of putting things in perspective, of course. But let me go to Debojyoti now, who's the geneticist here, and hammer him a bit, asking you about the fact that CRISPR-Cas9 has actually, and for those uninitiated to the fact of gene editing tools, it's one of the most powerful tools of splicing or cutting genes and replacing them with whatever factors we want to make a trait better or whatever we want to do with it. So CRISPR-Cas9 has revolutionized the way we actually are going to look into the future and how gene editing might make or break things for us, irrespective of whether it's human or otherwise. So let me also ask you the fact that we have had challenges apart from just the successes of CRISPR-Cas9, its limitations, and what other emerging gene editing tools do you see currently being in use, and how is that going to change our future?
Yeah, thanks for that hammering question. So, of course, we are no more in CRISPR 1.0, we are across that, we are in CRISPR 2.2, and, as more and more scientists are studying this amazing immune system in bacteria and now in archaea, we are realizing that there are more complexities that are emerging in terms of the systems that bacteria use. So we are really learning from bacteria all the time. So you asked about what CRISPR does and how to go on from there in advanced stages. So since there's a diverse audience, the CRISPR technology itself is like a word processor file. You have a mistake somewhere that you wrote in the letter. Let's say instead of writing you have written AHE, and that A is what causes a disease. That's the same thing that happens in the body. You can simply take your cursor there, press a backspace, introduce a letter, save the file, and that's exactly what CRISPR does. It takes an enzyme, cuts the DNA, incorporates a base or a DNA alphabet, and then this is saved because DNA replication happens. But there are other methodologies. One of them is, of course, where you could insert a change on the site without the need for a DNA break. And this, I think, is probably the most revolutionary technology. It's called base editing, and something which is more advanced is called prime editing, all coming from the lab of somebody called David Liu at Harvard. And this allows single bases to be changed with a chemistry of A to T, C to G, and without the need for a DNA break, making it much safe. The more recent ones include no more DNA editing, but RNA editing, which is because when you change something in the DNA, there is no way to undo it. So whereas RNA editing has this possibility that you could have the original RNA back. So RNA editing for some diseases, and somewhere in the horizon, something that also I am interested in personally in the lab is protein editing, where you could specifically change how proteins behave in a specific manner. So I think these are some of the areas which a lot of people are employed currently in, and also a lot of companies have come up trying to take them to the people. But I think you will touch upon this later on. Most of it seems to be very concentrated in the US market. So how and why is something we could all discuss about it. Yeah.
Sure, thanks for that insight. For me as a layperson, and for many of us perhaps, the scare is how else can that be used? And that's what we'll come to next. But first let me get to Subash, who's here representing personalized medicine, and how genetics is actually changing the game in personalized medicine, and how we are looking at individuals rather than masses of people for personalized medicine, right? And so could you give us a sort of overview of what's playing out in that area, and how exactly people should be looking at the ethics of all of this?
Sure. Hello? Yeah, so in terms of where genomics, so we've been in genomics for now 22 years. Just to give a little bit of a background, we started with a previous company called Awesome Biosolutions, and our focus there was research. So we used to work with research institutions and pharmaceutical companies. And genomics is kind of moved to the clinic, I would say, accepted in the clinic probably about 7 or 8 years ago. That's when we started really seeing, especially in oncology, newborn screening, rare genetic diseases, is where genomics has been accepted. And in terms of where we are seeing the industry move now is more towards personalized medicine, where it's an area called pharmacogenomics, where you're looking at your mutations and there are very clearly defined markers, and the US FDA has stated, asked pharma companies to actually state the markers as part of their submissions. And it's very well documented for about 150 drugs. And the idea there is if you have those mutations, either the drug could be toxic for you, you could be a good responder, bad responder, and the doctor can look at that and modulate the dosage and sometimes even change the drug. So that's the stage at which we are, that's what is being accepted right now in the clinic. Oncology, obviously, people are doing whole genome sequencing, looking at variants across the genome and trying to figure out if some of them is causing the disease, whether they can change medication or treatment plans. But it has taken about 20 years where we are coming to a point where we're using genomics in medicine and in the clinic. But again, the same point that you raised in terms of CRISPR, and like Debojyoti said, most of the research is being done in the US and China. And that, one of the things that we're also trying to address is that inequality. We call it genomic inequality. Whether it is data inequality, because there's very little Indian data out there. And that's a point that we've been with IGIB, we've discussed with SKB before, with various governments, that we need to get that data out there, available for researchers if we have to give that push for the industry to start developing new drugs, new diagnostics. And I think that is one area that, and I hope that with CRISPR and the current technology, that inequality doesn't widen. So hopefully, I mean, with researchers like Debojyoti, we will be able to bridge that gap.
So which basically brings us to the big question of how affordable the technologies will be and how scalable they will be. So let me just again open up the panel for all of you to comment on the economics of genetics and gene editing and technologies that might be informed by this science and take it to the next level. So the divide already exists, like we all have commented, but how do you see the future? Do you see this kind of technology becoming more and more available to people at hand, like they can order a kit from Amazon or something and do their own personalized medicine, which is available right now, but how affordable it is and how much money does it mean to, say, get one genome mapped?
So right now, I mean, so in terms of if you want to do a whole genotyping on the genotyping technology, the cost is, I would say, probably about $40. About 3,000 rupees is what you could look at 3 million markers across your entire genome. Whole exome sequencing probably is down to about 7,000 rupees. The whole genome is, I would say, about 50,000. So it's tremendously come down since, I mean, the early days when it was $3 billion, right? So in 20 years, it has really come down. In terms of affordability for preventive genomics, what we say is it's a once-in-a-lifetime test. You're looking at your germline markers, and those don't change. So we're saying once you get it, so you have to look at its benefits across your lifetime. You can always get a whole genome sequencing also done, then you don't have to repeat it. But I think the technology is quite affordable now, and and actually it's cheaper here, I would say, than in the US. Because of the US healthcare system, the insurance, the payer system that is there, the same whole exome sequencing that we probably charge 10,000, 12,000 rupees here is probably running about $1,000 in the US. So it's more affordable here. I think, I mean, it's a technology that large-scale adoption should come from the government or from insurance companies for this to become more used by people, and you can order a kit online and get it done. So it's quite inexpensive right now.
Yeah, so I think as far as genome editing costs and so is concerned, I think the devil is in the detail because it's not a machine-driven solution. Here we are talking about a cell therapy product. Currently, the ones that are in trials and have successfully, gone through trials are all based on cells that are manufactured, edited. So basically where a patient cell comes out, edited, goes back, or iPSCs which are edited. I think we have Dr. Indu here from LV Prasad. She is doing something similar with us for eye diseases and so on. So the cost at that level is very difficult to cut down because there is a cell therapy pipeline which is expensive because it's a good manufacturing practice oriented, and you're giving something to a patient from outside, which is going to be difficult. Where the costs would go down significantly, I see in the next 5 years, are where in vivo therapies will come in. The first therapies have come for liver because it's common knowledge that anything that you put in the body ends up in the liver. So it's easy to edit genes in the liver. But taking it to the next level where you could specifically give, let's say, nanoparticles with CRISPR in the eye, or nanoparticles going to the bone marrow, or nanoparticles going to the pancreas, that's the research as well as the final, let's say, cost reduction step which will come down, and I think both viral, non-viral vectors. So just like the last 10 years has been the age of CRISPR, I think the next 5 years would be the age of delivery vehicles for CRISPR. If that becomes good, then this would go ahead.
One very interesting part about costs are measured costs and unmeasured costs, so second order thinking, consequences of consequences. An example might be useful. Imagine for any reason, you sequence a young female baby and you find a mutation in a set of genes that increase the risk of breast cancer, right? There will be a cost of dealing with this information. The order of increase may not be a BRCA1 classical mutation where Angelina Jolie took a certain set of decisions. It's an increased risk. It might even be editable in the future. But there is no immediate risk. There is no way to even know at what time is the cost benefit beneficial, or in fact even the risk benefit beneficial of doing something. And who's going to handle this information? The cost will not be in getting the sequence. The cost will be in maintaining an ecosystem around that sequence such that further healthcare and responsibilities can be responsibly discharged in time to come. Let's imagine we don't even find the mutation. But we say only 30% of breast cancer genetic risk is understood. Which means every year there is a risk something will be found, the sequence has been done, and somebody will be responsible. So you need to effectively create that digital twin I was talking about, which exists in an in silico world, acted on by running algorithms every year, somebody to be identified as a responsible person for guiding the future care. So in a responsible health system, the hidden costs are astronomical. In an irresponsible health system that says we have sequenced you, here is the result, we don't have anything more to do with you, the costs are this much only. And given the increasing entry of the legal into the medical, all private players and governments, one would have to worry about courts, one would have to worry about other things. One must anticipate these costs to create a future health system. Just a thought.
Yeah, absolutely. I agree. And it actually takes me back to the times during the pandemic at its height. And Anurag, I don't know whether you remember, we had a conversation around the cost of sequencing and how much sequencing is much sequencing and who pays, and whether patient data can be anonymized and whether that can actually be sent into the big data banks of sequencing, you know, and used the world over, etc. So that's a great question, and it actually brings me to the ethical questions around editing. What are the things as scientists we are dealing with today in terms of why this hammer cannot be let loose to everybody who wants to hammer a nail, right?
No, I think that's a very important question, particularly in context of what happened in China with the CRISPR babies who were born, and no one knows about what has happened to them. But so I should make this very specific that it appears in media and CRISPR is so popular that and that there's a do-it-yourself kit available, I was told at some point. In reality, to do a CRISPR experiment and to take it to a successful completion is not something that can be do-it-yourself. So it requires a scientist still at the end of it. And if you're a scientist, then being a responsible scientist becomes a very important aspect. So I don't see this to be like a rogue individual coming and doing CRISPR experiments, changing humanity. That won't work. It would be a rogue scientist. So that said, what are the ethical things? I think we keep on debating about safety. We keep on debating about off-targeting, etc., etc. But for the parent, it is about adding 2 or 3 years to the life of the person. I mean, we get so many mails from parents who write that they have terminally ill children and they want to fund, and some of them actually are funding as well. And they are not really caring about when your CRISPR is going to be absolutely perfect to go and edit. They do not want that line to be drawn for their child, which is also at some point understandable, because this decision is really the decision of the family. So I think it's going to be very case by case. We would see more of N of 1 trials, sunshine trials being more common in the CRISPR field, and the really big population-wide trials would probably take a little bit more time, because there you have to deal with the legal, the ethics, and all these things.
So I think the keyword they were used was sunshine trials and things like that. It's always a risk versus benefit analysis. If you don't do it and things are guaranteed to go south in a way that a typical member of society would understand as unacceptable, that is clearly where CRISPR will find its way. Of course, people stretch the definition. If you look at what happened in China in Dr. Hay's idea, what he was supposedly trying to do was to introduce a mutation that would change the ability of the HIV virus to infect the T cell. It completely cures HIV forever, the introduction of this mutation. People have shown that if these people get a bone marrow transplant from somebody carrying this mutation, HIV is permanently gone. Now, his idea was vertical transmission between infected mothers, children, editing, putting it back. The reason we found it unacceptable as a community was it's not the only way. We can use antiretroviral therapy. Somebody who's passionate will say antiretroviral therapy has side effects. It is not 100% effective. And the logic analysis, how does this? But there will be examples in which there is no risk. And here comes a really critical part. There will be a black market for everything that people desire, are willing to pay to get done, but governments, laws and regulations say it should not be done. I guess the best example is drugs, right? I mean, you can find them, or even alcohol, sir, for that matter. These things can be found, and IVFs and stem cell therapies. So one must maintain that balance in which you don't penalize the one willing to follow the rules while allowing things to grow on a black sector, completely unregulated. And you would have to do appropriate public messaging. And that public messaging must be the center stone of all efforts to guide this clearly dual use type of technology scenario.
Yeah, I think I have a slightly different point on ethics and morality, and in that it's not static, right? So 100 years ago, eugenics was thought to be a great science, right? I mean, Francis Galton, I mean, all the people who got involved in that. And it turned out to be, I mean, what led to the genocide of the Jews. So I think, I mean, the responsibility lies with all of us in terms of with the governments, with the scientists, on how you enforce it. Right? And then there's a very interesting point in one of the podcasts, Dr. Dodner was talking about sickle cell anemia, how that can be cured, one of the experiments that are going on right now. And Miles Davis, who was one of the famous jazz players, had sickle cell anemia. And so the question was, did his music come out well because of the pain that he was going through? And if he didn't have it, would we still have Miles Davis out there? So very interesting questions. I mean, these are, I think, no easy answers to these questions.
Absolutely. Just like you mentioned, IVF was looked down upon when it was first invented, or sort of put in place. The Vatican, the entire, population in the West went against it. Same happens to stem cell. Last evening at dinner, we were having this fascinating conversation about how in the UK even, stem cells are still not the accepted therapy. And there's so much regulation around it, as opposed to IVF, which is found such cozy nooks everywhere across the country. So, yeah, lovely questions to actually open up the conversation to the wider audience. And let's see if we have some, there's a question there.
Thank you, sir. So I have a couple of questions. So first is to Subash, sir. Regarding the role of insurance, you were saying, so like my understanding is in India, it will be very tough to get reimbursement for genome editing. However, when it comes to whole genome sequencing and like genome sequencing as a diagnostic tool. There are insurance companies which might be willing to go take that role if that particular technique is proven to be cost effectiveness. And that brings me to my second question, sir. So that is on the, like you were talking about 2 or 3 different therapies, and which one is most common, which one is more cost effective, which one has most, which one is more safe. I am a health economist and we do this work, and there is a, so at Department of Health Research, there is a organization called STIN. So, and that particular organization is willing to take health, basically cost effectiveness studies for Indian public healthcare setting. So, I mean, in case if we can actually initiate talking with them and discuss about the role of, like, discuss about initiating such studies, we might get to see more of gene editing technologies or like into the Indian public healthcare system. And then the third question is on, to you, sir, is on the, like you were talking about RNA, DNA and protein editing. How, like, in terms of cost as well as effectiveness, how are these 3 different from each other?
I'll talk first about the insurance. So all due apologies to any insurance executives here. So Indian insurance is nowhere, I mean, covering most diagnostics unless you're admitted in the hospital. And then they're not even covering, unless you get admitted for a day, I think they don't even cover emergency care. So Indian insurance is way off. I'll talk a little bit more Western insurance, especially US would cover a clinical test that's recommended by the doctor as part of most insurance plans for cancer and newborn screening. What we are trying to do, and the interest that we're getting from insurance, is to do some sort of population studies. So ideally, I mean, they make money when their clients don't go to the hospital. So they're saying, can you say, I mean, like if there are cardiac risk markers in Hyderabad, we can do some preventive camps for their clients so that they don't end up in the hospital and insurance has to pay out. So that is where the discussion is currently with insurance. I don't think gene editing, there is anything out there right now clinically that, that can be reimbursed.
So as far as gene editing goes, there is nothing available in Indian market. The first FDA approval for sickle gene editing has just come out, and it's capped at $1 million. So that's the, as far as the cost is, goes. And they do not have any backup plan at this point of time to reduce the cost. We do not know how that would be when it eventually comes to India. We expect it would be much less, but cannot say anything about the insurance at this point of time. Yeah. You also asked about differences between the cost of, so I think RNA therapies based on antisense oligonucleotides or LNAs are already given as RNA therapies. And these typically are, I mean, it varies, but it's affordable to a lot of people. I know a lot of patients in India who get this ASO treatment. Again, not in India, but they go abroad and get this treatment. And some of them are covered by the Eli Lilly Foundation also for a lot of diseases.
Let me add a slightly different wrinkle to it, that I seem to specialize in that. There are 2 types of rationality when you talk about cost effectiveness at all. One is epistemic rationality, which is a true rationality. You'd be looking at the total cost incurred versus benefit to create to the patient. For example, an annual health checkup is epistemic rationality point of view useless for the most part. The second type of rationality is instrumental rationality. Rational behavior in terms of getting what you want. So when a patient visits a doctor and the patient is insured, the patient is already paid somewhere, they want something done. Doctor's instrumental rationality is patient satisfaction, and since they will also get something if something is done, so both people have an instrumental rationality of doing something, while epistemic rationality would say not cost effective. Insurance companies' instrumental rationality in the end is not about the epistemic. It's about can you make money off the model. So in the end, misincentives align with all insurances to lead to a spiraling cost of healthcare, which indirectly I think Dr. Bang was referring to with our new models of financing. So looking at insurance in isolation as a way of driving this while very useful for introduction of these things and making them affordable for at least a section of the population, requires further work by organizations like yours to look at the real discussion. This behavior is called rational irrationality, well described in voting behavior, typically occurs when cost to self from the rationality is very low, and one must figure out how to change that.
Hello, good afternoon everyone. So I have read it in news that China is, through gene modification, China is trying to build super soldiers. So is it possible, and my question is, what impact will it have on the rest of the world and on our environment? Thank you.
I have not read that news. I think it's still in the realms of science fiction. If you ask whether it is feasible, I think there are genes that can make muscles stronger. I'll only tell, just practically what is possible, not that it is essentially doable, but there are genes which can make muscles stronger, which potentially could give people the ability to pick up stronger objects. And there are genes which are possible to be mutated so that you don't feel pain anymore. So I think the combination of these two is already a lethal superhuman. So you can imagine that such kind of things is all there in the science fiction novels, but whether really somebody is pursuing that, I don't know. What impact? Well, it's like one of those Independence Day movies, no? You can imagine how it is going to happen.
It's okay. Sir, I have a basic question, sir. Sir, can everything be traced down at molecular level, sir? Whether it may be okay, sir, for Alzheimer's and Parkinson's, we can found exactly these reasons are for example, in Alzheimer's, we can say amyloid plaques are present. In Parkinson's, we can say dopamine levels. So, are other psychological problems like OCD or something can be anxiety disorders, can be traced down to genome level? Everything that's the first question, sir. Second thing, sir, we are saying that we want to have minimum side effects, sir. Sir, instead of changing the body, why not changing the environment stimuli? Because we are reacting to the stimuli from the environment and we are releasing those hormones. So why not only important, sir, it's important to study the molecular level, but changing the environment is also necessary, no, sir? I may sound somewhat this thing, sir, but I'm having that question, sir.
No, I think you make a lot of sense. There are some ways to understand heritability, the best of which is called twin studies. Twins can be of two types, identical and fraternal, right? Identical twins share 100% of genomes and fraternal will share by random chance about half. By looking at degree of inheritability of diseases and differences between identical and fraternal, you can estimate the degree of heritability. In mental health disorders, for example, schizophrenia has high heritability. Alzheimer actually does not have as much. Some types, of course, do. OCDs and many behavior disorders have even lower. PTSD, for example, has almost none. So it could come back to that. Now in terms of therapeutics, the reality is we don't know the genes. We know the heritability is high, but there is no single gene or mutations we can put our finger on for most things except neurodegenerative dementia disorders. So it's not an option. So the only way out actually is what you are saying. Counseling, environment, chemical environment, I can call them drugs. So in that sense, those are the only ways. Changing genes is not going to be the primary way of treating disease anytime in the coming future.
Well, as one other person has asked, we do have to worry about the implications for misuse of gene editing. He talked about the example of Chinese genetically modified super soldiers, basically. But of course, anyone else who's watched like sci-fi or something like that would also know about genetic enhancements, hints, and other problems. I'd like to know how much we'd have to worry about how the misuse of genetic editing in our current day and age through say the modification of existing genomes to create improved ones, but also we could consider how, let's say, the wealthy who would have access to such expensive gene editing could enhance their children by, say, increasing their muscle mass or increasing brain matter. So if we're not worrying about mass-produced super soldiers or biological weapons, what modern moral or ethical implications do we worry about the most?
I just want to have a round of applause for that question, because you could have sat here and done the moderation better than I am, but please go ahead.
So you can be certain that governments and defense organizations are looking at this. A very simple modification that is certainly being investigated will be fighting at high altitudes, where we know that genetic mutations exist that allow certain people to live better at high altitude. So clearly, without necessarily modifying the entire genome, one could introduce temporary modifications that could increase the capacity of soldiers, because carrying oxygen around is going to be more cumbersome. So definitely happening. The real question then, it comes back to the same thing, the black market and everything else I was talking about, is how do you build a system in which these things can be talked about openly? And the first most important part is to allow the discussion and not treat it as something that is inhuman or completely unethical, not going completely woke and saying it is not to be talked about. It will happen. The biggest challenge with allowing germline editing, and I'm on the foresight committee of WHO that looks at this, is today we say that race, ethnicity are not biological constructs, they are socio-political constructs. And all humans are equal. And in India in particular, with the caste system and everything else, these things are important. Now, if the affluent people could introduce genetic mutations that could be passed on by generations, making them stronger or fairer or anything, or better able to run at high altitudes or have greater anaerobic capacity, you would create that branch in the human race, and you can see authors, Sapiens and Homo Deus, already talking about it. One thing that you also know, now I'm going to plants, and the work of Nicholas Taleb, is anti-fragility. It is diversity that gives us robustness to infections. Let's assume that all of us decide to undergo a particular mutation with homogeneity on our T-cell receptor that prevents HIV. And we later find it allows a new virus to infect even better and kill us. Diversity is the strength that prevents extinction, and Taleb discusses it beautifully in his book Anti-Fragility. So for a variety of reasons, and I still believe it's not something that you should leave to governments and scientists. My own personal opinion, scientists are no better informed, domain-specific scientists are no better informed about consequences of their areas. Governments are even less. It is an educated public with a larger discourse that's going to do this. And young people will be the key part. So really glad to hear your question.
So I can add one point there, right? So the ethical problem already exists. So it's not a new ethical problem, right? In terms of medical treatments you have today, the access that affluent have versus a common man has, there's intercompany, there's intra-country differences. I mean, you saw it during the vaccines. The hoarding by the Western countries, not giving access. So the ethical problem exists. So if you want to solve it, I mean, let's not put it away to some distant gene editing problem. It's there today, and we're not solving it. So that will continue. I mean, unfortunately, that will continue.
I think the ethical challenge of a permanent enhancement of a particular families or groups is way, way bigger than temporary ones.
Yeah. I think there is a chance that you would have these differences in patients who are going to be treated faster, better in some countries than in other countries. And I see that will happen. I mean, there is nothing that can stop. but how the effect of what he said, the diversity in the genomes versus the robustness of a population is a matter of more of 100 years, 200 years. So not good for the race, but for a short-term thing which most of us seem to be always running after, this will happen.
Perhaps the good news is that just like AI, genetics and personal genomics is going to create a level playing field and bridge this divide at not such a distant future, where anybody who has the hammer, like we're talking about, is going to have experiments around the hammer, considering all the ethical questions that need to be considered. And as Anurag has pointed out, let's not just leave it to governments and scientists, have a more popular debate, discussion about these things. There's a question there.
So, I have a question, like the same scientist who was behind the CRISPR baby experiment. So he was imprisoned for 3 years, and in 2022, I guess, he was released. And now he's saying he will start his private lab where he will again use gene editing. But now he is saying publicly that I will not do it in germline. But what stops him from doing it in germline again if he's having his own private lab? He was doing this thing in a university system, but now he will have a private lab, and he's saying that he will get multi-million billion funding in a few years. So how can we keep a check? Should we not at least ban these who are like proved to be unethical? Or
I would take a slightly different view. Once somebody has completed their sentence, of course they can go back to life like anyone else. Number two, banning things, particularly those that you know will happen, simply drives things underground, and there is no ability left to discuss something that is illegal. We have made that mistake in a variety of areas, and I'll pick one that I think is terrible, and again, maybe different with psychedelics, for example. You will see in the next coming decade, a proper use of psychedelic compounds will grow and will become a routine part of medicine. It is inconceivable to me it won't happen. We live in a country that allowed their use in small quantities in different ways as part of things. And yet we do nothing for nicotine and tobacco. We speak on many other things. So the morality of these things, as was indicated earlier, is very complex. Banning things is not the answer. The only thing that stops any of us are two things: our own sense of what is right and wrong, and fear of rules that tell you what is right and wrong. And rules are only as good as the ability of a society to enforce them. Otherwise, all that you see is the biggest people get away with everything, others get stifled. So एक हिंदी में होता है कि ताला साहूकार के खुशी के लिए होता है, चोर के लिए नहीं, which basically means all these locks are for the happiness of the guys who think about these locks. The thieves are trained to break them. So don't trust in locks as your primary solution.
I'll just add that, banning creates heroes. So if you do something like that, there will be more gene quiz that will come out. And one fundamental thing to understand here is that germline editing will come. It's not that it's not going to be there. There are patient groups that are lined up to say that I want my embryo edited. Okay. It's just that the problem with the first set of experiments was that the technology was not well tested, foolproof enough to go forward, where there were alternate solutions available. I think that was the problem more. It is not about the fact that germline editing was done was the primary issue. So it will be done, and there is a lot of politics that is played over here when things come from China and so on. So we will not go into that, but I don't think so that aspect of banning would really stop this.
Absolutely. I guess what stops him from getting into germline is the phenomenon called government. Doesn't want to get into jail again, right? Once bitten, twice shy. I'll have to edit this session out now because we are running late and Anurag has to go catch a flight. So thank you very much for being such a wonderful audience, cozy, wonderful audience, and thanks for those great questions.